that has to be said is we wouldn't have this urgency if there weren't a lot of people really in big trouble around the world. It's still as frightening as it was 10, 11, 12 months ago, unfortunately. So this is where we are. When we discussed this webinar, it seemed worth having a quick background, and the whole thing started at Diamond, certainly when, well, I assume you all know what the main protease is about, one of the key enzymes that starts the reproduction. But it kicked off when Haitao at Shanghai, I believe it was Haitao, contacted either Martin Walsh or Dave Stewart and asked if Diamond could help because they had the structure and wanted to do co-crystallography. And it wasn't possible, if, as far as I remember, to get the samples to Diamond. So Martin's team knuckled down and recloned the protease. The sequence had already been known by then, obviously, and managed in massively fast time to get the thing cloned, expressed, and purified. In fact, I think the longest part of that was waiting for the DNA to show up from the vendor. And
yeah, that's correct, Frank. It was 2 weeks wait to get the synthetic gene, and we were lucky because already, you know, consumables were becoming hard to come by. So I think if we'd have been a day or two later, I think that could have, you know, John Grimes, when he got his polymerase synthetic gene, it took them 5 to 6 weeks, and he ordered it a few days after us. So we had a bit of luck there. Yeah, we had a bit of luck.
And so those Martins talked to me. Yes.
Just for the sake of historical accuracy,
please,
the thing that started it from my perspective was a phone call from Rao in Shanghai. So that was on the Sunday, the 25th or whatever of January, and then I talked to Martin, which started things, I think.
Yeah, that's right.
And I guess we'll start having less and less of the details as we go deeper because so much happened. But one more thing was Martin Walsh told me on a Monday, which was I think the 17th, if I'm not mistaken. He said, "We've got these crystals, they diffract to high resolution." I was on my way to my group meeting and I told my group that we should really knuckle down now. And then we finished all the experiments in 2 weeks. Got the data out very quickly. And so I think it took off from there. At the same time, Martin sent the protein to Nir's lab for electrophilic screening. And so what really happened, well, we had all this data and we knew it was going to come out, and UK was starting to lock down. And it seemed wise to just put it online the way that the Chinese groups had done. And we just literally put it on the website. Luckily, for the Fragalysis platform was mature enough to put it on there as well by that time. So it was available, and Martin sent out a tweet, which I think is what caught, maybe it was Matt or Alpha's attention, and they contacted Martin again. So this whole thing mushroomed. The other thing was, of course, it's important to say that because of the amount of data we had, we knew that the binding site was very well populated, and should give many opportunities for progression. And these are the figures that were in the paper that eventually came out. But one of the key things for me was this idea of these mergers. If you look at the individual hits, they suggested many compounds that could come out. And so this merge space we already knew was a powerful thing from a previous publication with Nir. We thought, well, there must be something lurking in there. So in my mind, this was the red herring that kicked it off. Maybe the others saw it differently, but this was how I was thinking. Because it seemed there was a premise that if it's such a rich compound, somewhere in this merge space, there must lurk the drug that will solve the crisis. Simple to make, potent antiviral, clinically safe. I'm saying a red herring because we know, well, certainly I know a lot more, and most people said it's more complicated, and it definitely is, but it still was useful in the sense that it got us talking, and the conversation in late March was broadly this. We were on the phone, Nir and I think Anthony as well, and Nir said, well, why don't you ask Derek to ask everybody to look at the data and and then submit it in a Google Doc, and Alpha then said they could do better, and they built this website to harvest the ideas. And John jumped in, John Chodera, and said, well, folding at home could help to rank them, and then we realized we could help with the assays in Oxford, and we also realized there would be a pool of philanthropic money we might be able to delve into. I think it kicked off at the Weizmann with Nir. So that's the background to all this. I think one thing is worth saying. Why did it seem useful? Because SARS had been around, MERS. Where are all the anti-SARS drugs? And I actually got this email from Al Edwards saying, the world doesn't need another protease inhibitor, but it wasn't in the clinic. So obviously people were looking for them. But I think one of the things that transpired, or that we agreed on in the following months, is that these existing inhibitors were largely covalent peptidomimetics, and which were apparently, if you look at the evidence, they were hard to get to be drugs. So there was an opportunity for a well-designed, easy to make compound. Yeah, so how did the whole thing take off? I think this must be well known now with the fragment screen. In the first instance, went out on a tweet. This tweet kicked off the ideation phase, which PostEra hosted, thanks to Alpha, Matt, Aaron. And then a lot of connections later just mushroomed into this very big effort that is worldwide. A lot of labs putting a lot of time into it, and effort, and pro bono expertise, and at cost experiments. And it's still running. That's the amazing thing here. I'll say this, though. What was really key, I think, is that right at the beginning, the moment Ed joined, he asked the question, what's the TPP? And embarrassingly, I didn't even know what it meant. But it really defined the endpoint where we were heading for. And that, I think, was key to just focus the minds. It's one thing to think of merge space or whatever, but this defined, there's an endpoint and there's a long way to go. And that has been our guiding principle from the beginning, since then. And so this is my last slide, just to say, where are we? Where's it going? The goal is, and it remains, a new potent antiviral, therapeutic and prophylactic. And we can talk at length of why this ought to be necessary, in terms of vaccines and everything else. But it does feel that the need for this will not go away. And the other point is that it should be simple to make and completely safe, and a good drug. The strategy about how to get it to the world was, well, if we work in the open, then ultimately other manufacturers would just pick it up. I'm sure there will be many pitfalls introduced by this decision, but it was unanimous. All of us felt it was worth doing this way. And the first phase, fragment to lead, these numbers are broadly accurate, although it all swims, but we got to the lead in something that we could call a pre-lead by September, early autumn. We're in the lead optimization phase and looking ahead to how we might get this to clinic. And that's what the next few presentations are about. We have a number of lead series. We've got a number of decent things, properties of our compounds. Ed will talk about it. And we need to think how we're going to get further. A lot of it we're making up as we go, as all of you know. But ultimately, thank you very much for participating and still participating. I'm going to head over to Ed now.
Thank you very much, Frank. Let me share my screen. I'm struck by a phrase that one of my colleagues on the design team used. He said, and I think he only joined the project a couple of weeks after me. He said, wow, he said, this is like jumping on a bullet train, isn't it? So it's been a remarkable team to work with, and I probably been about the best part of my year, but in 2020, that's not a huge achievement. So I'm going to talk a bit about the design team. I'm Ed Griffen. I work for a tiny business called Medchemica, and we do a lot of medicinal chemistry. The job of the design team is, I've described it as focusing the wisdom of the crowd. So there's a huge amount of really smart people out there. As of yesterday, I think we had 15,000 molecule ideas submitted, of which we've now made, we haven't, but the folks at Enamin and Assay have made about 1,300, and that's probably increased by now because structures come very quickly. What's the role of the design team? We provide input into the TPP, and the TPP is an evolving document. It doesn't stay still as we've learned things, it's changed. The test cascade, which I'm going to talk about in a minute. What I would describe as compound strategy, we talk about a lot, and we have been able to provide some input into protecting bizarrely open IP. So even that has required us to put some things on the website to say, no, really, you're not going to have this. We do a lot of prioritizing compounds for make and test, and the team has good cross connections into the synthesis group, testing groups, comp chem, and the structure generation teams. Most of the time we meet, there's actually about 20 of us. There's a lot of us with a lot of big pharma experience, which is very helpful because we've all been down the road of knowing what it takes to get things, not just to be ligands or even inhibitors, but actually things that can go into the clinic. Equally, we've got brilliant colleagues in academia who do stuff that amazes me still. So it's really a pretty good team to work in. We've already seen the TPP. It's important to understand that a drug is so much more than just an inhibitor. And Annette is going to talk about viral replication. And obviously, the idea is we stop the virus replicating in cells, but we don't kill the cells. Really what we need to do. We're talking about an oral agent because it's going to be widely used. That means you need a certain solubility and it needs to hang about. And that community use is what drives all these things. So you mustn't have drug interactions because patients might be taking other things. You can't be messing with their heart rate. You don't want to be causing genetic toxicity. You don't want to be causing issues with unborn children, because I would say when we're successful, our patient group will include a significant proportion of women of childbearing age. So these things are absolutely essential. So we keep all of this in our heads pretty much all the time. This is a brief view of, we do design, synthesis, and then we get into answering these key questions. And there are assays run literally globally. Some of this has been run by collaboration with pharma, some of it has been run with CROs, some of it has been run by excellent academic collaborators, and we're pulling it all together, and we've got this far. So the next picture, everything in Navy, we've actually done. So we've been all the way into rat PK. The hamster may or may not be the best whole animal model. That's one of the challenges, is it's not completely clear. We might go with mouse, which would have just different issues. But in order to get to here, obviously you've got to have oral availability. There's no point in going into animals until you've got enough potency. And we want to look at what's happening in cells, both in terms of engagement and antiviral assays. And it's a cysteine protease, and there are plenty of proteases in the human body. So we have got some protease panel screening, and that's looking particularly good already, which is a huge relief. And you need to know about plasma protein binding because you need to do the scaling when you're understanding this, because in the end, we want to predict our doses to human. If people are not aware of it, all of the data is stored in a collaborative drug discovery database, which is available via the web. And if you haven't got access at the moment, then you can just get in touch with me as one of the administrators, or Lisby, and we're happy for anybody to look at this data. It's very well organized, it's very well curated, and you can line it up and look at it in your own time. The thing I would say is this is a flexible critical path testing cascade. So not everything on the previous diagram is on here at the moment, because when we haven't found a problem, we don't test in it, but we are going looking for where our problems are. So you would expect to see this change. We'll be doing some hERG screening. We'll be doing some Ames testing as compounds develop, but we don't run everything all the time. Okay, I'm going to just give you some snapshots. Some of this, many of you will already know. The aminopyridines have been one of our key series from the get-go. We had this tiny little compound composed of just joining two fragments, already 23 micromolar. We've made more than 300 of those now. You can add simple groups to it, and that's reaching out into P4. You can see the structure there, and we get almost a 10-fold improvement. Or you can push out across into the prime side, and you can get another 3 micromolar, and that's good. Or you can improve your P1 group, and you can really make a real step jump in terms of potency with this isoquinoline. And of course, inevitably, you start putting those ideas together, and we're down at the kind of 250 nanomolar level. So that was about a month ago. That was where we are. There's going to be an update in a minute when it gets even more exciting. Oh, that's a shocking piece of build, because here it is. What if you take that good idea and this good idea and you combine them together? Does the SAR actually combine? And we were thrilled at the beginning of this week. This is a ligand. This is not a drug. But what it's teaching us is that if you go P1, P2, P4, P1 prime, we can get down to that kind of 50 to 60 nanomolar level that we'd had to. This isn't an optimized compound to any degree, but it tells us what's possible. We need to fix this. We need to fix this. We know we can improve on this. So what it's telling us is we can make better compounds. Huge step forward. Another way we can do it is we can actually evolve that left-hand ring. Here we can get a doubling in potency. It actually turns out that if you just pull out the single enantiomer, you're down at 140 nanomolar. So you can see from these things that the SAR is beginning to build to make it more credible that we can get the really potent compounds. And this is really exciting data that we've had through collaboration with the Israel Institute of Biological Research, thanks to Nir and the team at the Weizmann. Actually, viral activity in a cell line, SARS-CoV-2, and key here that I want you to take away is the drop-off from enzyme to cell is only about tenfold. And that's reinforcing Frank's point. This is not a peptide mimetic. We actually know that this is a highly permeable compound. So when you haven't got too many hydrogen bond donors and acceptors, you can get into cells and you can stop the virus replicating. So that was another real milestone in the last 9 months of work. Just gathering together some of that data now. So August, December, our goal, well, we're getting close in terms of ligand properties. Solubility could be improved, protein binding, really pretty respectable. Across several antiviral assays, we're seeing consistent potency, but not cytotoxicity. A really clean protease selectivity panel. I talked about this is NDCK, MDR1 is permeability. Clearance, a bit high, but I'll come back to that in a minute. And we've got measurable oral bioavailability. It's low, it's not what we want, but when I show you some of the clearance data, I think I can persuade you that we're going to be able to work on that. We have another series. So this is a series of quinolones that originally come out of the SARS, you might describe as COV1 work, where they weren't able to optimize them further. We've already now been able to take this series down below the micromolar level. We didn't have activity in cells when we were at this level, but our previous experience would be, as we drive the potency down, we should be able to get the cellular activity improved. Again, very clean against protease. These are not particularly permeable, but we have ideas about how we can improve that. There's some pretty obvious things to do first. More stable in terms of clearance and better oral bioavailability. Still not enough, but we're on the board. And that's, again, the thing that counts. We have a kind of final backup series, which is benzotriazoles. These, again, we've seen some SARS-CoV-2 activity. We're down at the half micromolar level now in these compounds. Some concerns that some bits of this structure we might want to fix, but the fact is we're making progress. They are currently quite strongly cleared. We'll see how they do. Again, I think we've got some actions that we can take to mitigate that clearance. But one of the things that's very clear about these is how they're coming out into P4. And that's kind of exciting to be able to work on that. Okay, I said that there are things we can do with clearance. So this is all of the data I'm showing you is measured. There is no modeling in what I show you. This is human liver microsome. This is solubility data. We've had this data, I think, about 10 days. And here's the benchmark compound that is relatively highly cleared and not really soluble enough. And we've done the methyl walk around it. And if you put a methyl in this position, making this a quaternary center, you can drop the clearance and you can improve the solubility. Alternatively, you can replace that oxygen with a nitrogen. There is actually an amide version as well, which is also good, or you can replace the whole ring system with one that's 1 carbon smaller, and you can make real improvements to both solubility and clearance. So these are obvious hybrids that are now in our plans. What are we doing right now? So the team at Enamine have been working extremely hard because we know that this vector is one that we can build out from towards the P1 prime side to build into potency. And it looks like also those quaternary compounds may be more stable in terms of microsomal clearance, increased solubility. We have a range of libraries that we're currently working on. John's going to pick up how these have been prioritized later on, but this is one of the places where we're working to improve all the properties at once: solubility, potency, solubility, and clearance. We think we can attack through this kind of strategy in our lead series. And I've hopefully not overrun my time, but that's to give you a brief snapshot of how the design team's working to prioritize the current targets. Thanks very much.
I think it's now my turn to take over. I'm Annette Von Delft. I work at Oxford University as a translational scientist, and I wanted to talk to you a bit about what happens with all these compounds that the Medchem team is designing all the time. So first of all, I just wanted to give you an overview of where we are at the moment. So Ed has mentioned a lot of the assays that we are doing already. So Ed is working right up here in the compound design, and Tatiana at Enamine is helping with the synthesis. And a lot of you on the call today are contributing to this part of the project. What then happens is once the compounds are made in the Ukraine, they're sent out immediately to Nir London's lab at the Weizmann, who are doing a fluorescence assay. Sorry, Nir, I switched them around. And the Oxford group is actually doing a fluorescence assay that is performed in Chris Schofield's lab at the moment. And the compounds also get sent to Diamond, and the hits from the fluorescence assay and the rapid fire are attempted to put into co-crystal structures. We're also running some target engagement assays at the moment in Oxford, and Nir is setting them up in Israel as well. And then the compounds that perform well in these two assays, we select to send out to some of our antiviral collaborators. And I've seen some of the collaborators on the call. A big, big thank you to all of you who have been running these very work-intense experiments. So we've got people at Oxford, in the Netherlands, at Radboud University. We've got people in Israel who are running assays for us at Imperial College. There's Patrick St. Reed in Nebraska who has been running assays for us, and also Nir, this is Nir Drayman, another Nir who sits in Chicago. So a massive thank you to all of you, because we really, really appreciate to have this direct readout that gives us information of where we have to lead, where the Medchem strategies have to go. Then next, we also try to establish whether these compounds are not only effective, but also getting into the cells, making it into the body, and hopefully having a profile that doesn't interact with body enzymes. So when we're looking at CYP inhibition, for example, these are liver enzymes that if you target them can cause some severe side effects in humans. So you want to have compound that is metabolically stable and safe, but also where you kind of have an idea of where the safety profile is going to sit later on in the in vivo experiments. What we are currently trying to set up and haven't got completely established at the moment is what we'd ideally have is a compound that not only targets SARS-CoV-2, but potentially other coronaviruses as well. We have some collaborators that we are talking to at the moment that have these assays set up in their lab, although we haven't yet sent any compounds out to them. The other thing that we would like to see is that these compounds are not causing any viral mutations. So for that, you would run serial viral passage assays, which are extremely work intensive. And we're talking to collaborators about that at the moment. If you know of someone that has the capability to run that and may have some spare time or effort to do these assays in collaboration with us, we would be incredibly grateful, because as you may have realized, this whole project has been run on goodwill so far. We've come incredibly far until now with that, but to get to the clinic where we want to be with a compound, we still have a way to go. So to move the compounds through this kind of assay cascade, we want to achieve certain things. I'm not going to go into these into detail, but what I want to say is, first of all, a thank you to the groups at Chicago, IIBR, Radboud, Nebraska, Imperial, and Oxford, who have been running all these different assays. We appreciate that it is these are really difficult and work-intense assays, and you have been working through lockdown and BSL-3 in the BSL-3 facilities to make this work. Thank you to you all. I sometimes get asked, why do we run so many assays? The answer to that is mainly we've got a lot of different compounds to profile, and we use the antiviral readout to guide our medchem strategy. So we've been taking any offer from any collaborator to run a compound for us. And so far we have been very, very lucky to actually have people running these assays for us. We have different cell lines, very E6 cells, the virologists tell me are very easy to grow and enable a quick readout. But we also have the group in Oxford using color 3 cells that provide a more physiological readout, but are difficult to grow and to handle. They seem to be the prima donnas in the cells. Also, the other thing that we're looking for is some redundancy between the different assays that we run. So some of our very best compounds we have been sending out to different labs to get an idea, because as you can see here, the techniques that are used to establish antiviral efficacy are very variable across the different labs. Just to give you a taster, this is one of our lead isoquinolines that has an efficacy of around 100 nanomolar in biochemical assays. Nir ran this compound in Chicago using the virus OC43 and hasn't seen any efficacy for this compound or an IC50 over 20, whereas the collaborators in Israel ran the same compound and saw an IC50 of 1.57 micromolar. This is wrong. This is needs to be the lab in the Netherlands. They performed the same. They performed a qPCR assay on SARS-CoV-2, so that is this line here, and they saw an IC50 of 200 nanomolar in cell culture. So what we don't know at the moment is why do we see these widely varying assay readouts? We know that it is quite difficult to reproduce data across different labs. When we look at publications on, say, compounds like remdesivir, we get quite a wide range of IC50s from different labs. But we also do know that our collaborators are using different strains and different timings in their assays. And with this particular compound, we think there may also be a solubility issue to it. So that is one of the things that we still have to improve on our current lead series. So these kind of varying readouts is the reason that we would like to see antiviral efficacy under 1 micromolar in 3 different assays across 2 different cell types before we actually go into any further investigation and animal efficacy assays. We want to see all these other things as well. So a non-toxic compound with good solubility, a good metabolic profile and oral exposure that will give us certainty that we are on the right way. And this is a slide that you probably don't even need to read through. It basically just shows the complexity of what we have ahead of us if we want to get one of our compounds to a clinical trial. So this is called IND, Investigational New Drug. We've got a whole range of safety assays that we have to go through. There's a lot of logistics involved. How can we actually make the compound at the quantities required? And we have to kind of go through some very stringent dose range findings and tox data for this. So that was it from me.
Thanks, Annette. Oh, yeah, Nir's next.
Hey, can you see my slides?
Mm-hmm. Yeah.
So hi, everyone. So I want to start by saying that I was lured into this.
Nir, we can't hear you if you're talking.
How about now? Yep.
Hearing? Yes, I'm hearing Nir.
Okay.
Yeah, I can hear him.
Okay.
Good.
So I started saying that I was lured into this project by the promise of inhibiting the protease covalently, which is the focus of our lab and our scientific passion and our expertise. And in a bit of trickery throughout the project, as you saw the compounds that Ed and Annette are pushing into the clinic, we lost our covalency. Which is very sad, and I'm trying to get it back. But originally, this part was supposed to be about the assays we're using. So our original assay that plugged into the screening that Frank mentioned and that sort of started this whole process was an electrophilic fragment screen, in which we screen our library of fragment electrophiles against, in this case, the protease, and then read it by mass spec. And here you see the mass spectrometer we have from one angle, but this is how it always looked from the other angle. And this is Efat, who's responsible for all the screening in this project. And here's what the typical result may look like. In blue, you see the mass of just the protein, but when it's incubated with compounds, you can see a mass shift because our compound always bound irreversibly. And so they increase the mass of the protein, and we can know which compound bound. And we discovered a lot of nice covalent compound this way. But then, you know, the people with 20 years of experience in pharma said, oh no, covalency is a liability. We need to go reversible. We tried to find our place in the project, and pretty soon we realized that there's at the time, we didn't have access really to good activity assays. So Ronen here took upon himself to develop a fluorescence-based assay for the emperor protease. And the assay is pretty simple. We have a substrate peptide that the protease recognizes, and we have a fluorophore on one side and a quencher on the other side. And if the protease cleaves right after this glutamine, then the fluorophore is released and we see fluorescence. And this is how it may look on a plate reader. So this is accumulation of fluorescence over time. And if it's only the enzyme on itself without substrate in blue, or in orange with an inhibitor that inhibits it, you see the disappearance of fluorescence. So this is one assay that we're basically basing our medicinal chemistry and design strategies to sort of improve the potency of inhibition in this assay. In parallel, the Chris Schofield's group in Oxford are working on a completely orthogonal assay. Their readout is not fluorescence, but rather mass spectrometry. So they're following the actual masses of the full peptide, which decreases as the protease cleaves it, and the product peptide, which is smaller in mass and increases as the peptide is cleaved. And so each compound is being tested in both assays. And here you can see a correlation, pretty updated correlation of all the compounds tested in both assays. And there are some outliers where one assay or the other claims that these are artifactual compounds and we should not follow them again. And every time such a compound arises, we have heated discussion of which assay works better. But for the most compounds, you see a very nice correlation validated in 2 orthogonal assays. So this really gives us confidence that we can take these compounds further. And so after we optimize the initial condition for this fluorescence-based assay, we stepped a little bit aside and let the real experts take it. And from there, almost all the work at the Weizmann is done at the Nancy and Stefan Grand Israel National Center for Personalized Medicine and is headed by Dr. Chaim Bar, whose office is right here in this beautiful building. And Chaim's team are real experts in automatization and high throughput screening of various assays. And so I just wanted to introduce you to the team. So Galit Cohen is responsible for all compound management. Each week we're getting a plate with compounds from Enamine, and these have to be reformatted into dose response plates that are suitable for plate readers. And Galit is doing all of the compound management, reformulating, and is a key person in allowing us to run this assay so fast. Then Shirley is one of the people that actually runs the fluorescence assay and makes sure the results will be on the CDD on time for the design team to make it happen. So I tried to couple each photo to the CDD assay protocol and name that they ran. So we also have Hadir, who's also running the fluorescence assay here. And this is Leo, who helped us. So one sort of offshoot of the Moonshot was a high throughput screen performed at the INCPM of 200,000 compounds against MPRO. And this allowed us to identify some new compounds, which are not sort of our main lead compounds, but pending their structural determination may give us some hints on how to further optimize our lead series. And Leo was instrumental in running this high throughput screen. And then, as Annette mentioned, we recently imported from Professor Susan Baker this nice luciferase, cellular luciferase assay, where we have cyclic permutate luciferase that has as part of it this sequence that is recognized by the SARS-CoV-2 protease. And if the protease cleaves after this glutamine, it allows the luciferase to complete itself and it starts generating luminescence. And if we inhibit the protease, then we don't have luminescence. And so we can assess the activity of the compounds in cells. And the Oxford team are doing one copy of this screen, and now it's Weizmann. Sylvia is also started running this team and using the capability that we have for high throughput screening. We think we will be able to screen hundreds of compounds maybe in this assay, and maybe salvage compounds that maybe are not the most potent in vitro, but in cells are more active than some other compounds. So I think that would be very useful. And helping Sylvia is Noga, that is also responsible for running cytotoxicity assays that we're doing in parallel to Enamine. And then lastly, not as per se, but we do support some of the chemistry in the project. So Ram and Paul from my lab helped with the initial chemistry, both of some initial inhibitor series, as well as Ram synthesized the first fluorescent quench peptide for the protease assay before we started outsourcing that to a company. And Christo is now working on high throughput chemistry to develop some combinatorial chemistry that Ed didn't mention, I suspect because they have covalent origins and he's an anti-covalent person. And so they helped a lot. And lastly, I want to introduce Daniel from my lab, who's the father of this Oogie series that I mentioned that was born in sin out of a covalent inhibitor, but is now converted to a reversible inhibitor and is also showing some promise. And Daniel also provided a lot of computational chemistry support at the beginning of the project. And I'd like to end by acknowledging the rest of my lab who may have not contributed directly to the project, but have suffered immensely from me being involved in the project. And so did not get a lot of attention over the past year. And they're very accommodating and provide moral support for everyone else. So thanks everyone, and thank you for your attention, and everyone I might have left out. And I'll give the stage to John, I think.
Well, was it
Alpha's
Alpha
next.
next?
Yeah, I can start.
Oh, Alpha, sorry.
Hey all, Alpha here. Thank you so much for the support to Moonshot. So I'm Alpha Lee, a researcher at the University of Cambridge, and also a co-founder at a startup, PostEra. My academic research group and also PostEra works on applying machine learning to accelerate medicinal chemistry, specifically in this project trying to accelerate and augment chemical synthesis. And we work very closely with the really talented team at Inamine and Assay, who has been amazing chemistry wizards in churning out a lot of compounds very rapidly for the project. A great pleasure to work with such an amazing synthetic chemistry team. And in the next few slides, I will talk about how we have used machine learning to augment the chemistry efforts in this project. The underlying technology that we developed over the past few years is a technology that allows us to rapidly predict the outcomes of chemical reactions and therefore solve the reverse problem, which is how do we rapidly design synthetic routes to organic molecules and therefore triage organic molecules based on how easy it is for it to be made in the lab. And the underlying idea there is an idea inspired by machine learning, in particular natural language processing, which is the idea that there are a lot of published chemistry reactions out there that we have harvested from patents. Why not parameterize a model that inspired by natural language processing can learn to predict the product given the reactants and reagents. And in summary, this is literally a natural language processing problem because we can convert reactants, reagents, and products all into a chemical text, which then can be fed into a machine learning algorithm. And over the past few years, we have benchmarked and developed this algorithm to an extent that outperforms quite a lot of the machine learning approaches out there in the literature. And actually in the recent paper, we showed that it can match human performance and therefore augment chemistry expertise. And this algorithm was applied at the onset of Moonshot when we got a lot of very enthusiastic support from the community. Thank you to all who submitted compounds to us. Actually, in the first week alone, we got over 2,000 compounds submitted based on Frank's and Nir's and amazing fragment hits. So the question we immediately asked ourselves is, well, we have 2,000 compounds. I mean, theoretically, we can go through them one by one, but we don't want to spare the enemy team having to do so. So how are we going to pick out quick wins from the many amazing suggestions that we have received from the community? And that's where an algorithm, unlike humans, can just do by working around the clock. It's just some GPU time. And in fact, we are able to triage 2,000 submissions in the first week to 200 compounds with less than 3 steps of synthesis in less than a weekend. We are able to rapidly identify compounds that are rapidly makeable. For example, the compound in the bottom right that turns out to be a single-digit micromolar binder is only a 2-step synthesis, and we weed out compounds that unfortunately were too complicated for us to make in a short time. And you may ask, well, why there are many sort of algorithms out there that can quantify synthetic accessibility. But I think one force multiplier that we have is the amazing work done at Inamine, which has stockpiled over 200,000 unique compound building blocks, and with many, many more made-on-demand building blocks. And therefore, we realized that we can actually make a lot of really complicated and interesting and highly functionalized molecules easily if and only if we can keep track of all the building blocks and its reactivity. Although we have a lot of chemistry insights, keeping track of all these building blocks and keeping track of what is makeable from this vast chemical space is something that an algorithm, again, is uniquely able to do. And that's, again, another avenue why machine learning becomes incredibly important in force multiplying the synthetic effort, because we are able to rapidly decide which compounds can be made using this amazing inventory of building blocks. And just give some examples of richly functionalized building blocks that we can find. For example, this compound has been made using the route in the top. Once it reached the roadblock, manifold platform can identify another route starting from a richly functionalized building block is actually only a one-step synthesis. Another example, the route on top again starts from a building block, which might be the best building block at the time, but we immediately realized that there are actually other building blocks out there, which allows you a more richly functionalized building block, which allows you to realize the compound a little bit quicker. I think through these iterations, we're able to force multiply the amazing synthetic chemistry efforts that we have been receiving at Assay and Enamine and the CROs that we are partnering with. We are also starting to deploy generative models in combination with synthesis models, and this was an experiment done in July showing that we can use generative chemistry to design compounds with some activity, and showing how we can use this to explore scaffold space and understand what the protein tolerates and what the protein doesn't. This technology has been since then deployed at a large scale on Manifold, which is a product marketed by PostEra to offer to drug hunters beyond the Moonshot initiative, where this platform allows people to synthesize and search across all available molecules. Just go to postera.ai/manifold, and we have created the world's largest chemical space to make and order compounds. This is directly integrated with the world's top CROs to get your compounds made. In short, enter the compound that you want made, and we would be able to rapidly decide and triage whether this is something that is already in stock or rapidly synthesizable. And if it's synthesizable, we can rapidly order the compound for you, get it made and shipped to your doorstep. That's the end of my part. I would hand it over to John, who will talk about another piece of amazing enabling technology, namely computational simulation, which has allowed us to progress at rapid speed.
Thanks so much, Alpha. Here we go. So I'm going to talk a little bit about, so I'm John Chodera. I'm at the Memorial Sloan Kettering Cancer Center, and I'm lucky enough to be involved in the organization Folding at Home, and part of the Folding at Home Consortium, which is a consortium of laboratories around the world that run a distributed computing platform. Really thanks to all of the volunteers that donate their computer time. Many of them are gamers, and so when their GPUs aren't cranking away on playing a computer game, they're donating their compute cycles to run molecular simulations on multiple different platforms, and now we even support Raspberry Pis. But thanks to these volunteers, we had accreted something like 100 petaflops of computational power. And at the beginning of the pandemic, we thought we would try to use that power to help in whatever way we can support our experimental colleagues and collaborators in different ways. And so we posted a blog post early on, this was February 27th, saying, we were trying to understand the nature of antibody binding and the evolution of resistance, and just see what we could do to help our experimental colleagues in multiple different contexts. And something entirely unexpected happened. Due to sharing of this blog post around and then making it to popular media, we ended up with more than 1 million volunteers coming on board, and finding soon we had the first exascale or exaflops computing platform in the world. Much larger than any of the supercomputers that you could fit into a single room. And there's a huge number of people I can't hope to thank. Some of them are listed here in the lower right as part of a preprint that also reports this amazing computational advance. But we thought we would really try to use this computational power that had been volunteered from all of these folks around the world to do what we could. And this is when we got in touch with the COVID Moonshot to see if we could help them in multiple different ways. So the different laboratories, and I can't go through all of the contributions here, but the Bowman Lab at Washington University, our lab at MSK, Vince Voll's lab at Temple University, tried to figure out how we could help each of the different stages as the moonshot progressed from initial hit compounds all the way to potent lead compounds in these more final stages of drug discovery. I'm only going to talk about the last part, these relative free energy calculations, where once the Medchem team from Ed and working closely with the PostEra folks with Alpha was able to come up with a synthetic route or synthetic routes that they liked that would be simple, quick, robust, and produce a large number of compounds from a given intermediate that explore in a particular direction to try to engage here, for example, in the P4 pocket. They could enumerate very large lists of compounds that we could potentially synthesize very quickly, but sorting through those compounds is challenging. And certainly the med chemists can pick some compounds to explore the SAR, but wouldn't it be great if we could use our computational power to jump straight to more potent compounds? So hence this concept of running sprints to try to identify which of the compounds we should actually pick and buy and try to make as we engage these different pockets and growing out from our main scaffolds was born. We use a technique called alchemical free energy calculations, and it's called that not because we're transmuting lead into gold like the old alchemists, but because we're changing one molecule into another through a pathway that can't possibly happen in real chemistry, but it can in a computer. And this is a thermodynamically rigorous method that gives us the total free energy of binding within the approximations of the force field and the protonation states that we've chosen for this calculation. And the way this works is that we start with one of the X-ray structures from diamond. And I can say that you know, having the availability of crystal structures so rapidly after the compounds are synthesized is the only way this can possibly be incredibly successful, because we have this beautiful structural data to start with. We can enumerate all of the different conformations in which this proposed molecule might bind by sharing the scaffold interactions that we want to preserve. We select the one with the best docking score, and then we run these non-equilibrium free energy calculations to try to compute the free energy difference. And retrospectively, this seems to work quite well in capturing the overall free energy changes for compounds that we had already synthesized. So we were very encouraged by this in going into our first sprint. I have to thank people who have really contributed to this. William Glass is now supporting this effort from the UK. Hannah Brees McDonald did all of the work to set this up based on code written by Dominic Rufa in the lab, who's a graduate student. And then Matt Whitman helped us deploy this at scale and really summarize some of the data in these cool dashboards I'll show you in just a second. So we decided to try to focus the folding at home community around these sprints where they would really try to devote as much computing time as they could to help us get answers to the chemists as quickly as possible. So we set up this front page that allows us to summarize where we are in each of the sprints through a progress bar. It's also appearing on the PostEra website, so you can see where we are. We've just finished sprint 5, which I'll mention in just a moment. But it's amazing how much computation we chew through. These are not the typical free energy calculations where you just run a few nanoseconds. These are very substantial calculations where we are making very large transformations compared to what's traditionally done on these kinds of free energy calculations. Instead of adding a couple of atoms, we're adding maybe a dozen atoms and maybe subtracting a dozen atoms at the same time. So these are challenging and really pushing the boundary of what's possible in these free energy calculations. For example, in this first sprint, we chewed through 10 milliseconds of simulation time, 10 milliseconds. Now we take femtosecond time steps. So this is a very large amount of computational power that ends up going into this. Just to show you what we were doing in this case, sprint 1 focused on trying to identify a replacement for this potent but problematic beta-lactam group over here. We had hoped to recapitulate the 7-fold increase in affinity, and we did pick a set of compounds that we predicted by free energy to increase the potency. And it turns out we found some that were pretty good. Our best compounds were only 2-fold worse than the beta-lactam ring. But unfortunately, they're all also quite lipophilic, which is a challenge that we're trying to overcome now by making more soluble compounds. So we're still trying, and we'll come back to this, to come up with better engagement in this P4 pocket. One of the interesting things that's come out of this from the earlier sprints is that most of the ideas we chewed through were much worse. They were mostly bad ideas, and there's a very small tail of good ideas here at lower free energy, negative compared to our reference compounds. And what's also interesting is that the human chemists do a very good job of proposing potent compounds, but it's hard to get Ed to recommend 10,000 compounds that we can make. So their distributions are shifted, at least through the predictions, towards much more potency. But just because we can chew through so many more compounds using this automated method, we end up hopefully proposing more potent compounds this way, at least through the predicted model. In our sprint 2, we aim to optimize again the P4 pocket through a different chemical route. We ran into some issues with the chemistry there, so we didn't end up with too many of those compounds. Sprints 3 and 4 focused on another promising series, this UGI series that Nir mentioned before. This is still an amazing reaction. To me, it blew my mind that you can install so many different substituents in a single step, one-pot reaction. And so we've been searching through multiple different substituents simultaneously in this case. And some of those ended up with reagents that were sent then to Nir's lab. Sprint 3 was benzotriazoles, I should mention. I don't have an update on that one yet. We're still analyzing the data to see how well we did with that one. Sprint 5, which is the latest sprint that is just completed, builds on this very potent high ligand efficiency scaffold. And it turns out that we have a couple of structures from Diamond which very nicely overlay that we could build on and show just slight differences in the protein position. Ed enumerated the very exciting path through multiple different intermediates that are being made in quantity. And we're actually pursuing 3 different strategies here to compare how well the free energy calculations do against a simple pharmacophore model. And of course, the awesome Medchem team's picks for compounds that will actually push us forward. So we've set up a science dashboard, which is letting us helping us track the front runners here. We have a leaderboard of different compounds. We can also examine the reliable transformations because not all the calculations converge. We make it possible to download the snapshots, and now we're pushing these snapshots to Fragalysis as well for interactive visualization. And all of the data is going on to the AWS Open Data Registry. And this data will be very useful to many others in the future, understanding how to analyze free energy calculations. So we're excited to start the next sprint. And at this point, I think we're happy to take your questions and the Moonshot team will be able to answer them, hopefully to the best of our ability. All right.
Anybody have questions?
You could either post them in the chat or just raise your hand.
So any of the speakers or only to the last one?
Oh, any of the speakers.
Oh, yeah, well,
Any of the
Anything.
speakers.
Any
So
question you
I'm
might
not
have
sure who.
Yeah, any of the
at
speakers.
all.
Okay, because the last one was just impressive. But so the target for the drugs are always the same pockets, right? Or is there a possibility to think of combination therapy that yours? I guess we're now aiming for one magic bullet, and that makes it easy to generate.
I don't think we're aiming for a magic bullet. I think in the end, everyone recognizes that antiviral therapy will very likely be a combination therapy. At the moment, we within the Moonshot only have the capacity to look at one target, so that is the protease. There is a lot of data coming out based on fragment screens on other targets that we potentially follow up. That will be hopefully picked up by other people or developed within kind of a different context. But I think at the moment we're going for this target. The future, let's see what the future may bring.
It's difficult enough, I guess. And another question about the antiviral assays. So I saw that there was, I guess we had a very, I guess the data that you showed was in our lab was very potent, and in Chicago it was not. But that is, I noticed, a different coronavirus. Huh?
Yeah, that is entirely true. So Nir, who's also on the call, offered the OC43.
And so how similar are the proteases at that pocket?
They are very similar. I don't know the exact homology, but I think they're over 90% homologous.
Okay. Yeah. But I guess it could explain that big difference, that there's no potency in that assay. Yeah, I guess for the antiviral assay, we may want to discuss this in a smaller context, but I guess I saw also CPE as a readout, which is probably much less easy to inhibit. I guess a CPE as a readout will be an infection that takes longer than we would harvest. And so I think it is probably a more stringent assay, but I'm not entirely sure. I think we should sit carefully and compare the assays.
Ronald, I should add about, you asked about the other coronaviruses. In an ideal world, we would have got those enzymes cloned and assays set up, the in vitro enzymatic assays. That's actually some conversation with funders that keep asking for that, obviously. Right now it's out of scope, or it's out of what we can embrace. But obviously the complexity, the other question is, if enzymatic inhibition is equivalent, why would the viral inhibition not be the same? And then you got the discrepancy between labs and all that rubbish. So it's clearly a big hairball, this whole thing. But
yeah, I think the current aim is we'll do as good as we can on the issue, which will be assessing different cell types and hopefully also assessing different readouts. And hopefully if we have a compound that really is very antiviral and does something, it should show up with a kind of consistent readout across the different assays.
I can comment on the OC43 assay. Hi, this is Nir from Chicago. I'm sorry I'm late for the call. So first of all, it's not a CPE assay, it's an immunofluorescent assay. So that I think is equivalent to what the other...
I think the other, so I think the IBR is doing a CPE.
Yeah, IBR.
Oh, okay,
doing CPE SARS-CoV. Yeah.
And I think, the proteases are definitely very similar, but we've had cases, for most of the antivirals we've tested in general, if they work for OC43, they work for SARS-CoV-2. But we definitely have many outliers for that. So one of the really well covalent inhibitors for the SARS-CoV-2 protease, GRL0496. It inhibits SARS-CoV-2, but it doesn't inhibit OC43 at all. So there are, differences, obviously. And I think that will be important when you think of what this drug is going to be developed for. If it's going to develop for SARS-CoV-2, then you don't care about the other coronas. If this is going to be stockpiled for future pandemics, you want something that is much broader.
Yeah, I agree. Thanks.
I see there's a question in the chat. I'll just start with it. So James Tufts asked, are there studies on the variability of the 3CL probe based on different strains of the virus? Is it highly variable? I think one reason people like it as a target is because it does not seem to be massively variable. I don't have the numbers in my head, but it seems to be one of the more conserved parts of the genome. The last time I looked, which admittedly is quite a lot of months ago, I couldn't spot in even the genomic tables of the viruses across the world, the COVID-2 viruses, that there were any mutations in this virus. But obviously, the moment you start poking at it, it will pop off and mutate. There's Sod's law. So I think the other question that we have discussed, but don't really know how to attack, or we don't have the bandwidth, is if we can make sure we target only the apparently non-variable elements of the binding site, so that it can't just quickly mutate away with a site mutation. We'll see.
Functionally, I believe the protease cuts in about 10 different places, so it's hard to imagine, or not hard to imagine that there's a lot of selective pressure to keep that invariant.
Yeah, I should add that a great point is that for the 3 series we mentioned, the UGI, benzotriazole, and quinolone series, those are chemical probes or hits for SARS-CoV-1, and even the IC50 against SARS-CoV-1 M-Pro translates very well to SARS-CoV-2 M-Pro. And so we have been very fortunate to be able to stand on the shoulders of those giants and continue the series forward.
John, do you want to take the next question? I'm laughing because it's an active conversation in our inbox right now. What's known about the enzyme kinetics and the coupling between monomers?
This is a great question. This is something that we're trying to dig into, both in terms of what's known from the literature and what we're seeing in our data. But certainly there's some interesting biophysical results we have on direct binding that differ sometimes substantially from the inhibition data. And this is something where it's been proposed that the two active sites are asymmetric or not equivalent in the sense, or at the same time, they're not necessarily both active, or they may be in different conformations. This is sort of supported by some of the simulation data that's been collected as well by multiple different groups. And we're actively looking into whether this means we need to revise our models and interpreting all of this data simultaneously. And David Min has been generous enough to help us think through this problem.
So Ben Perry, general question, what would the presenters single out? By the way, you're all welcome to contribute. Single out as the main bottlenecks in the current process, not taking away from how fast it's gone. I think not, I'd say not knowing what the runway is. You know, it's not being able to strategize further than a couple of months ahead. And that's because the funding, obviously, you know, the money involved gets bigger and bigger as we go. So that's sort of personal stress levels for what it's worth. Yeah, maybe others should comment, please.
I have a perhaps controversial take that I think humans are the main bottleneck here. We have some fantastic humans working on this, but it's also quite exhausting. In the first parts of this project, we were meeting every day to try to set up the strategies and move through to the decision cycle. And it can be exhausting, and it does slow our response to things like pandemics. So there's a lot of considerations, of course, in drug discovery, but wouldn't it be amazing if we had some sort of autonomous reasoning platform that could do this kind of thing, at least on some well-worn targets like cysteine proteases, to allow us to respond to future pandemics much more quickly. The med chemists may not like that idea, but I think the idea of enabling them to manage many more projects with much less cognitive load sounds at least appealing to me.
But sorry, can I jump in? Because the question was about the current process. The other one that I feel sort of a mental fight is the general assumption outside of this project that there will not be a drug in time for this pandemic. You know, it is not worth trying because the timelines are so long. And so every time you talk to anybody, that comes up and is the main assumption. And this may be correct, but the other point is, when will that shift, and what will it take in the world to shift the, let's say, compare medicinal chemistry to vaccines, which one has failed, which one has succeeded? Well, right now, vaccines are much better at doing this than med chemistry, even though we know so much about medicinal chemistry around the world. So it's an interesting dichotomy which we're up against. And every time we talk to a funder or anybody else, it's like, there's a long way ahead, and this is true. So figuring out whether we can even get through that and go much faster than it has done without the massive resources that the company has, I think that's what I feel is an obstacle.
Frank, there's an easy response to that, because, you know, if you just look at SARS itself, and if we, you know, back in the early 2000s, if we put a couple of $100 million or pounds into this, we would have saved the world trillions of pounds and dollars. And, you know, so as you pointed out, and others have pointed out, the beta coronaviruses are really highly conserved in the main protease. So if we get something that's going to, you know, bind strongly to SARS-CoV-2, then it should work against SARS-CoV-3, SARS-CoV-4, you know, and, you know,
and MERS and others.
Yeah,
it was really,
exactly. You know, MERS starts to diverge a bit, but I think the active site is conserved. But, you know, so it's really, really important that we continue this, because we need antivirals, as everyone in this Zoom call knows. So, you know, I don't think it's that difficult an argument to get across. But at the moment, all the focus is, as we've seen, has been on piling money into vaccine development. And there does need to be some kind of shift towards supporting antivirals as well.
And I had a really interesting point on exactly this point, Martin, from the science head of PHE, so Public Health England, who pointed out that at that big WHO meeting about 6 years ago, this exact point was made, that we will need a small molecule inhibitor against SARS viruses at some point. And that point was dismissed at the time. So I hope that this pandemic will give proof that we shouldn't stop working on this just because we have a vaccine.
I think it's going to be hard to ignore it, considering the amount of deaths and you know, infections we've had. With SARS and MERS, it was kind of contained very quickly, and there was a small impact on public health. But this virus has been absolutely catastrophic. Yeah, so I can't ignore it any further. But you know, we have to push hard, because it's the same with antimicrobial resistance. There's still this kind of you know, no desire to really put huge amounts of investment across the globe to try and actually combat antimicrobial resistance as well. So these are real serious issues that we.
So I've got a question from Vladas here to everyone. Yes, we have been communicating with clinical trial scientists for phase 1. we have a facility here in Oxford that is interested in taking up a molecule that may come through. We have talked to NIAID in the US, who seemed interested in helping us with the preclinical development. And we've got colleagues at the DNDi that we're talking to, and then also a colleague at Liverpool, William Hope. So yes, we're talking to people that seem interested not only for phase 1, but also for the kind of larger setup of phase 2A. Or yeah.
Can you say something about whether or not we need the in vivo efficacy model?
Yeah, that's a good point, John. That was something very interesting when we talked to the NIAID last week. They mentioned that there may be good scope to skip the in vivo efficacy model, because from what we currently know, it doesn't necessarily represent or replicate human disease on the kind of level of lung pathology and even viral load clearance. So they suggested that at least the FDA in the US may be interested in taking a drug forward that shows good antiviral efficacy on cellular level in combination with a safe profile without seeing efficacy data. So we'll have to see where that goes, this communication, but there's certainly an option to move fast on an antiviral and even skip the in vivo efficacy.
The most surprising part of that conversation to me was the need to show that these compounds don't interfere with current standards of care, even though the standards of care don't do much. Remdesivir, for example, is approved for emergency use by the FDA, even though it may not really prolong survival.
Yeah, that's been kind of shown to have no actual impact at all now with that recent WHO study. So
no, but the regulatory point is that because it is an approved standard of care, you need to demonstrate that it doesn't interfere, even though it doesn't do it.
Even if it doesn't work.
It isn't of any use. This is the point. You know, it's just making money for a drug company. So, John and Alpha, like from the point of view, the computing power and the stuff you're doing is amazing. And you know, one of the the other thing, of course, is loads of things that you have to do or you're doing now. But there was also this idea that we could try to disrupt the dimer interface as well. And is that something that, is going to take up a huge amount of your CPU and GPU stuff? Or is that something we could, because the original hits we had, they're not very informative. They're on the surface and stuff, but clearly we've got some indication that disrupting the dimer could actually inhibit the enzyme as well. So any thoughts on that for people in the call?
The concern from my end is always that you have to pay the penalty of several kcal per mole to disrupt the dimerization interface because it's in the micromolar range, presumably. So, we thought we'd fight the smaller battle first and go with the active site inhibitors because we know what things look like that bind to that active site. And of course, the fragment coverage was just absolutely beautiful in guiding our concepts about what kinds of interactions we're trying to make. And Frank's original hypothesis about how these make a roadmap about how you can really jump to potency was really realized in how people were able to do this in their heads. So we should be able to recapitulate this computationally. I love the idea of inhibiting the dimerization interface, but it seems like we have to bring in another, either be very, very potent or bring in another trick, like maybe Nir's covalent inhibitors if there's a cysteine nearby, or some way to point it to a degrader or something like that to have a weaker compound be able to get by.
I think also at the level of pragmatism, it's quite a leap of faith to invest masses into something that still needs proof of principle. And I think out there, I'm not aware, people correct me, that this premise has yet got strong evidence that it works as a general way of doing it. Well, I'd love to know examples. Email me, please.
There's also other fun things you can do. There are these rapalogs in cancer where they take two crappy drugs and link them together with a flexible soluble linker. And so you could imagine that if you did find even a moderately potent dimer interface inhibitor, just by linking it to an active site inhibitor, if you could get it to reach along, then it might be much more potent because of local concentration effects. But then you run into all sorts of other problems by having a gigantic molecule.
Yeah, it's all very interesting, and obviously it's kind of diverting from where we need to be at the moment, but it is interesting. Yeah.
So there was one point in the chat that I also wanted to pick up from Wall Ward. which kind of says the onset of the therapeutic effect for small molecules or for antivirals is much faster than for vaccines. And this is something I'd certainly underwrite. We'll have prime boost regimens mainly for the vaccines that are on the market at the moment. So we'll still have about 35 to 40 days until you have sufficient immunity. And there will be circulating virus for a while, especially with the pandemic like we have at the moment. So I still feel that there's a strong scientific case to push for small molecule inhibitors.
Does
The other thing that worries.
Go ahead, Frank.
The other thing that worries me is, all those people that with the vaccination won't work, and given so maybe the vaccines will suppress, create herd immunity, but it's a worldwide thing, and the vaccine has really got everywhere in the world. A lot of people will still die, and it feels like it's going to be years out there.
Well, I think it's going to take a while to vaccinate the world. That's for sure.
Yeah.
So,
especially with the cold chain requirements.
Yeah, exactly.
But I mean, it will be a multi-parameter game. It will be vaccines plus small molecules plus neutralizing antibodies for the really sick people. Like it's not going to be one road. And I think what we still need to convince the funders of that this is worthwhile going after, and the other part will hopefully just come together with the work. I think that's all questions. Right?
Are there other questions out there? Okay. I just had one more bunch of slides. I guess the wrap-up was formally thrown at me for some reason. So I'll just put up one more thought about this whole business, which I think.
Can you unmute yourself and I'll mute myself again?
Fine. So sorry, I'm just trying. I just need to find my slide. Apologies. And maybe it ties into this question of what can we learn about this effort as a whole. And 1 second, sorry. Share my screen. These are talking points, possibly polemical. You make of them what you like. But I mean, a lot of people are trying to do philanthropic or some sort of charitable clinical discovery. There's a general assumption that's always there that you need to do some kind of return on investment funding of these efforts, otherwise they won't get anywhere. There's always an assumption of a market. And the question is, why is the market failing? Fewer people seem to be asking, is there a market at all? And what do you do if there is no market? And this is maybe paradoxically one of those cases where nobody thinks it's worth making a drug, i.e. there's no assumed market. Discuss. Anyway, what I learned from this is if you don't define an endpoint, you'll never get close. That's something most people in academia, if they haven't worked in drug discovery, won't have in front of their eyes. I certainly didn't. So it's very much a project management question. I've been struck by this. The funding has been impossible to raise from conventional funders, certainly none with any urgency. You'd have thought if it's a long lead, I need to fund it soon. Just, as they've been joined up thinking, even in the UK, we've got all these drug discovery units. Maybe the government should have given each of them 10 million to begin with and say, off you go, guys, get working. It's just been sort of out of category. The other thing about this is if you're certainly new to the game, you need to have very diverse expertise at the very start. We were lucky to convince a bunch of people that was interesting enough, and Ed pointed to the team. This is not at all a given, and I think it only would have happened in a pandemic of this urgency. So does it scale? Could you do this for other diseases that people have accepted as part of life, even if they kill millions of people? TB is a good example. What would generate that urgency? Because the expertise clearly is there to move fast. And the other thing that, I guess, on the flip side is what we are drawing on is decades of experience and investments from the industry, which is, of course, funded on a return of investment principle. So definitely there's, if you like, indirect payback, but it's quite indirect. But the next one is, it's not clear the business models for this sort of thing actually exist. So that's why it seems worth going. It's worth trying. The other thing that the firmer the advice in this game, I guess, the more it should be ignored, because if that means a lack of honesty about how much we don't know. And I'm talking about the business model question, not about can you get a drug. And then finally, there's a huge technological gap that we can point to even in this project. You know, the amount of hours that John and co had to stick in to get those calculations, the piles and piles of hours that and days, months that Inomines chemists have put into just synthesizing this stuff. You think, really, can this not be shrunken down over the next decade? And maybe projects like this, I don't think we can claim that this project itself will do it, but projects like this maybe can help shift the funders' mindset about things that won't have a big market or where the market isn't obvious. So maybe that is a longer term output, even if we don't manage to convince someone to get to clinic or whatever, you know, phase 1 trials, or the compound fails, because compounds do fail, obviously, and that just happens. So I think these are what I think, looking beyond the particular urgency, which is vast, obviously, other things are urgent too. So we'll have to keep wondering about this, we as a community. And I'll keep saying this, in the race of vaccines against medicinal chemistry, sorry, med chemists, the vaccines won. And there's something out there in terms of assumption space that needs shifting. And I wonder what it will be. If there aren't, I see another comment flash up, another, which I can't read now, because I'm not Zoom competent. What was the comment?
Is it worth costing?
So John Spencer asked, is it worth costing the whole?
Yeah, that's an excellent question. You know, because we got our sort of cash outputs, which we can quantify, and then the in-kind effort is vast. I don't know how to cost it. Well, it is a good, excellent point. You know, what is the inputs in terms of intellectual effort, hours, pain, frustrations that we don't even have. Well, certainly I have little way of having visibility of, because it's been such a dispersed, anarchic, in the true sense, project. So what would you have to do if you wanted to fund this drawing together of experience? I don't know. It won't be I think it's about 600K we're at now. It'll be much more. And so the technological gap remains vast. I think I will then take the privilege of just calling it to a close. Thank you, everybody. It's been a wild 2020. This one maybe is a little bit of a light point in the really crappy year. So all of you, I hope you have a good break and get some rest, because we'll need it.
Happy Christmas, everybody.
Happy Christmas.
Let's hope next year is going to be better than this year.
Oh, very worse, can it?